Now, to understand the life history, what I want to do is to explain a little bit from a conceptual point of view the body of theory that defines how we predict life histories should evolve. To understand this, what you have to do is to think of your life as if it were a pie, and you divide that pie up into different slices. One of the very big slices of your pie is maintenance. It's the energy and resources that you put into replacing all the different parts of your body that are constantly wearing out and that are in need of repair or replacement. Another slice of the pie goes to growth. Many animals will invest some of the resources that they're consuming into getting bigger. A third component of the pie is storage often in the form of fat. Now, you may have a hard time thinking of fat as being an adaptive feature of your life history, but that's because we're inclined to make more deposits than withdrawals. For most organisms, fat, in fact, is a very important feature of the life history. And the reason is because resources are not always abundant. Fat is a way of establishing a savings account, of taking resources when they're abundant and saving them for a later point in time when they're not abundant. Finally, the fourth slice of the pie is reproduction. Now, there's a body of theory that predicts what the best way is to divide up the pie among these four different functions as a function of the kind of environment in which the animal is found. And so I study the life history as an adaptation, as a way in which the animals fit the template of their natural environment. In particular, I'm looking at how the life history evolves in response to the risk of mortality. Now, life history theory, which existed and was well developed before I began my work on guppies, predicted how life history should evolve in response to mortality rate. And so what I did early in my research was to make the connection between predation and mortality rate. If you live with a predator, you have a higher risk of mortality. And in fact, early along in my research program, I figured out how to measure mortality rates in nature and showed what you might expect. The guppies that live with predators have a much higher mortality rate than guppies that live in low predation environments. Now, what life history theory predicted was that animals that suffer higher mortality rates, that in those circumstances, natural selection will favor those individuals that attain maturity at an earlier age and that invest a larger slice of their pie into reproduction than into other things, and into, for example, growth or maintenance or storage. Now, what this means is that you don't do this for free. By maturing when you're younger and putting more into reproduction, you're taking resources away from other things. And so what life history theory really does is look at the optimization of how those resources are allocated. Now, an early ambition of mine when I started doing my research was to take a body of quantitative theory that made predictions and to test those predictions in a natural setting. And so that's why I chose to study guppies in Trinidad, because there was a natural setting, guppies that lived with and without predators that suffered higher or lower mortality rates, and that paired guppies with a theory that looked at how animals should evolve in response to differences in risks of mortality. So that theory gave me predictions, and I could test those predictions. The first way I tested those predictions was to compare guppies from natural high and low predation environments. I collected guppies in nature and preserved them and brought them back to the laboratory where I dissected them and measured variables that characterize the life history. I also did genetic studies in a lab very similar to the one that you see behind me, where I evaluated the genetic basis of the life histories of guppies from different types of environments. Now, what I found is illustrated in the cartoon that you're looking at right here. What I found in brief is that guppy life histories differ in a way that's consistent with theoretical predictions. The guppies from high predation environments are younger and smaller when they attain sexual maturity, and here you're looking at the smaller mother at the top of the slide. But these smaller mothers begin to have babies when they're younger, and they make more babies, and the individual babies are smaller. These mothers also put more of their available resources into each litter of babies that they reproduce, and they reproduce more often. And so at the outset, what I found is that guppies from high and low predation environments do indeed have different life histories, and the nature of those differences are highly consistent with predictions derived from quantitative evolutionary theory. What you're looking at here is what the guppies actually look like, and the point is that the differences are not subtle ones. They're differences that you can see if you have a collection of fish in hand. The left-hand column is a group of mature males from a high predation locality, and the right-hand column is a group of males from a low predation locality that was just upstream from the high predation locality. So when we talk about differences in the age and size at maturity, part of what we're talking about is the evolution of body size, and the evolution of a difference in body size is big enough for you to plainly see. Now, this pattern that I found in guppies between high and low predation environments, and the physical structure of the environment where I found them enabled me to fulfill my lifelong ambition. And that ambition was to do an experiment on a natural population in which I could test the predictions of a body of evolutionary theory. Okay, here you're looking at a schematic of the experiment that I did. What I did was to treat streams as if they were giant test tubes. And the way I did that was to take advantage of barrier waterfalls that create discontinuities in the distribution of guppies and their predators. Now what you're looking at here is a schematic of a real stream, and what's illustrated by number 3 is a high predation locality. This is a locality where guppies co-occur with predators. Stream number 1 is an area of stream that's above a dramatic barrier waterfall. And this is a barrier waterfall that stops all species of fish except for the killifish, Rivulus hardi. People often ask me, why is it that you can find rivulus in streams where no other fish are found? And the reason is that rivulus are the ecological equivalent of a salamander. On a rainy night, they'll happily jump out of their stream and hop around the forest floor, and they'll penetrate and invade aquatic environments wherever they can be found. I found them in water-filled tire tracks in the middle of muddy roads. I found them in depressions in the forest that filled on rainy nights. And in fact, I've gone to such depressions during the dry season and have dug down into the leaves, into the wet leaves that were in the bottom, and found rivulets sitting there, waiting and hoping and praying for the next rain to come. But the main point is that we very often find portions of these rivers above dramatic waterfalls that have only rivulets. Now this created the opportunity to do one type of experiment, and that was to take guppies from the high predation site below the barrier waterfall and introduce them into the previously guppy-free site above the barrier waterfall where only rivulets can be found. And what this did was to create a new low predation locality. It moved guppies from a site where they suffered a high risk of mortality and let them establish a new population in a site where they would have a low risk of mortality. So I'd use the source site, the founder population, as a control and compare the control to the introduced population and see how they changed relative to one another over the course of time, and in that way be able to evaluate how guppies evolved in response to low risks of mortality. Now, in the section of stream that's number 2, I was able to do a different type of experiment. In some rivers, what I found is that there are barrier waterfalls that exclude predators but not guppies, and further barrier waterfalls upstream. So in this case, what I was able to do was to take predators from below the barrier waterfall and introduce them above the barrier waterfall and let them extend their range to the next waterfall in line, which is usually about 100 or 200 meters away. And what that means is that the guppies that lived in that site now had a new increased risk of mortality, and I was able to see how they evolved in response to that increased mortality risk. As a control, what I did was to look at guppies that lived above the second barrier waterfall that would limit the upstream distribution of the predators. And so what this means is that I've been able to either decrease or increase the risk of mortality in natural populations of guppies. And in that context, I was able to ask, can I see these guppies evolve? Do they evolve as predicted by evolutionary theory? Do they evolve in a fashion that's consistent with the differences that I see in guppies from high and low predation environments? And furthermore, does that mean that I can actually quantify the rate of evolution in nature? Now, an interesting thing is that when I proposed doing this in 1980, the response that I often got was a wan smile, and somebody would say, you know, that's a great idea. We hope you live long enough to see something happen. It turns out that I didn't have to wait very long at all. I didn't begin to look for differences in life histories until 4 years had passed, and it seemed to me a little cheeky to even think that something would happen in 4 years. But it turns out that in 4 years or less, a lot had happened. The male life histories had evolved to the predicted endpoint. The introduced males, the ones that were transplanted from high to low predation, for example, now had life histories that were typical of males from a natural low predation environment. The females took somewhat longer to evolve. I didn't begin to see female life history differences until about 7 to 11 years after the introduction happened. But what I was able to establish is that the life histories do indeed evolve as predicted by evolutionary theory, and they evolve at a remarkably high pace.